So what we're going to do now, we're going to take about 15 minutes for Q&A. What I would ask, because of the time, is if you could limit your question to just one per person, that would be much appreciated. And we do have mic runners, so if you have a question, if you don't mind, just raise your hand. I'll point to you, and then we'll bring a microphone to you, and just please wait until the microphone is in your hand before you ask your question. So we'll start right here. Go ahead, and if you don't mind, you can stand up and ask your question.
Thank you. Very interesting. Interesting. Thank you. Is that working?
Test. This one's working.
Testing?
There
Is
you
that
go.
better?
There you go. Perfect.
Thank you. Probably a question for Dr. Rose or anyone up there. So many interesting different therapies that are being evaluated right now. My question is, are there any early studies in using multi-therapies together? Like, is there a science behind using gene editing, gene therapy together in animal studies at this point?
So, great question. The question was, just to repeat it, the ability to use multiple potential treatments together, gene therapy, gene editing, neuroprotectives. At the moment, the ability to actually combine experimental treatments is limited if it is a biologic or drug, but for delivery devices, it is possible to put an experimental delivery device along with an experimental treatment. That's really where things are happening, like delivery of the gene therapy vector that Dan talked about, the AAV, along with its package. The FDA is looking at potential abilities to do experimental in two experimentals at the same time. It's a bit cumbersome at the moment, but I know from discussions with FDA when I sat on the cell and gene therapy panel, they are looking at potentially how to do that going forward.
Excellent. All right, next question right here.
With respect to Luxturna, you mentioned a variety of risks involved for those patients who are taking the drug, but you didn't give us the number of those situations that could occur by taking the drug. You didn't mention, what the extent of those risks that came about were.
So the question was basically how prevalent were some of these safety factors, and for the best way to really look at that, I don't want to take the time to go through all those, is to really look at one of those prescribing information inserts, and that will give you all the actual numbers. Now, some of those are just relatively common to intraocular procedures. We did have a very small number of what we would call serious adverse events. So for the most part, we had a relatively good safety profile, and I think that's probably the best way I could answer that from here. I'd be happy to follow up with you later and get you one of those inserts, and that gives you a lot more details on that.
Do you have a time period over which you are going to be monitoring the patients who received the Luxturna medication to make sure that the improvements are going to last?
Yeah, so right now there is what we call a long-term follow-up, and that goes for about 15 years. We also are implementing a safety study on patients that are receiving this as a prescription drug, and that will also be for a 5-year period from time of injection.
Now that there's been a development path developed through Luxturna's success, is there a plan to actually roll out, using that same development path, actual treatments for all of the common mutations that have been identified?
I'm sorry, it was using the same path as Luxturna for
So
other.
basically taking the next most common mutations and then packaging them in AAV, delivering them to the patient cohorts that have those mutations?
Well, I think currently there are many other groups that are looking at the different genetic diseases for inherited retinal disease that are using the AAV technology. And one thing about the AAV technology is there's so much safety data, both preclinical and now clinical, that they can really leverage some of that. I think, though, it's not just looking at the common mutations, but what is actually workable, because each mutation or each gene has its own characteristics, and so it's not necessarily just numbers, but it is what we can actually accomplish doing development on that gene.
Okay, go to the back.
To lux levels, but is that expected to maintain and the disease progression is going to stop at that point, or not sure yet?
So I think that is one of those questions that we still will look for in time. And the one thing about Luxturna that we have seen is that there's been a restoration of function. So, and that is may not necessarily be the outcome of all gene therapies. I mean, gene therapies, there can be several outcomes in the sense of you're either slowing disease, halting disease, or you may actually recover some function. And so in Luxturna, that's what we've seen, is that we've actually increased light sensitivity. We've also improved in other visual parameters following that. But for the others, there may be a success, may be just halting disease or slowing progression down, because some of the diseases are much slower in developing than RP65 disease.
Okay, we have a question over here on the far side. While we're getting a mic over there, just a reminder, if you have a question, go ahead and raise your hand. We've got maybe about 5 more minutes that we'll take for questions, and then we'll go to the break. So we'll come over here to the far right.
Hi. In reference to the NACA study, one of the endpoints was OCT, and I think from understanding the rest of the talk, OCT is the mapping of the retina. Is that the endpoint of one of the main endpoints of the NACA study, the small cell molecule study?
Yes. Yes. Thanks to the FFB group getting that as an endpoint, that's replaced visual acuity as the endpoint for our trial.
Very good. We have a question right here.
Good morning. Great information. I had a question about the folks whose photoreceptors are basically, in my situation, I believe mine are about gone. I was with Shea Eye Institute under Dr. Jacobson. And I quit going because, the test, they couldn't really test me anymore. I couldn't see well enough. So is it what's the best approach currently, the thinking for dealing with people whose photoreceptors are gone? Is it stem cell implantation, or what's the future in that direction?
So as I talked about, there are a couple of approaches for those who are without any photoreceptors left, so there's no gene therapy or gene editing that's going to work. There's a session on optogenetics with Dr. Flannery. That is one potential area, turning the remaining retina actually into a new type of photoreceptor and using the neural network that's still there in order to put a signal to the back. And obviously, as I said, the stem cell work, i.e. regrowing the retina. The optogenetics, there are already 2 clinical trials ongoing, one in France, one here in the U.S. in Dallas. The cellular therapy, as I said, there's an ongoing clinical trial. I think it's going to be a question of which one shows the promise early enough to be able to go forward. But the direct answer to your question, sir, is that there are 2 potential options for people who have no vision left at the moment: optogenetics and cell therapy.
All right, very good. We'll take maybe another 2 or 3 questions. Right.
Dr. Chung, could you share a little bit about when a patient receives the Luxturna treatment, where are there specific locations where they need to go to get the treatment? Is it an outpatient kind of procedure, or are they in the hospital for a couple days? And how long after they receive the treatment do they see some improvement?
So I can only, so the question was where are these being done currently? So there are about 8 or 9 centers that are being set in the United States, and we can give you information on that as to where those are. Because this is not a clinical trial anymore, you know, for us, we had a very regimented way of seeing each and every subject. Certain times, what you're going to find is now it's more part of medical practice. And so there are different regimens, for the most part, at every center. But these are trained vitreoretinal surgeons and IRD specialists that see retinal patients. And so they have their own regimens, and they have very good follow-up that way. So each center might be just slightly different. Maybe you're seen on day 1 or day 2 or something like that. But then they will have a standard, you know, follow-up procedure that is applicable to them. Most of these, from what I believe, these centers are doing outpatient surgery. But I can follow up with you offline for a few more details on that. Plus our prescribing insert as well. We'll have those details.
All right, very good. We'll take 2 more questions and then head to the break. We'll come right here. Yes.
Thank you. Because NACA is not commercially available, do you recommend taking NAC? And this is probably a question for Dr. Rose. Do you recommend taking NAC, and at what level, or what dosage?
So the question is, since NACA is still in clinical trial, the original study by Dr. Campachero was with N-acetylcysteine. The reason for adding the amide is for more bioavailability. So here's the rub. When analysis has been done on NAC that you get at GNC centers or other over-the-counters, the amount of N-acetylcysteine in them is extremely variable. It is not really up to standards. So there is only a particular set of suppliers that are not over-the-counter that would be applicable for taking NAC. I would suggest that just going to GNC and getting NAC is not going to do you any good. We've run into that with other supplements as well. TUDCA is one that the foundation supported research in for a number of years. So bottom line, don't believe what it says on the label in the supplement stores. And as a matter of fact, it's unclear what real amount is in there, so nobody could tell you what to take as far as that amount. And secondly, without a control situation, I know some people who've tried it and say that it has no effect, but then again, without particular measurements, you're not going to know.
All right, very good. And our last question in the back left part of the room.
If someone participated in a previous trial for something such as Luxturna, does that prevent them from participating in a current trial or a future one for stem cells or something similar that might add to the benefit?
So an interventional trial of a gene therapy or a cell therapy, usually, in most cases that I've ever known, are rule-outs for other clinical trials. Whereas a treatment like NACA, which is a drug that will wash out of your system in a certain period of time, is less likely to be a rule-out for a future clinical trial. A permanent intervention, such as a gene therapy or a cell therapy, essentially is a rule-out for any future clinical trial. However, if something becomes approved, then it's up to the physician and the individual to decide what to do going forward.
All right, very good. Thank you very much. So just in conclusion, well, yes, please, big round of applause for our panel. Thank you very much. And thank you, everyone, for your attentiveness. This is a great room, a lot of energy, and it's amazing that there's this many people here. We're going to go to break now. Just a reminder that if you are on Twitter or you're on Facebook, you can participate in the conversation using the hashtag VISIONS2018. And the next session after the break, we'll be learning more about our Legacy Society program, and then we'll be back in here for our keynote at lunchtime. So thank you very much, and enjoy the rest of the programs.